Alzheimer's disease and other neurological diseases that have amyloid protein as one of their signatures are really difficult to study because in patients you see these huge plaques, but that's the endpoint of the disease, it's not the beginning.
But what if amyloids can be seen early on?
Maybe we can actually design therapeutics that actually can attack the disease at a more basic level before too many cells are killed, before you see the amyloid plaques in a CT scan, for instance.
And that's the challenge. Matthew Liu says amyloids are so tiny they can't be visualized using conventional microscopic techniques.
You have to label them with a special type of molecule that'll glow. And with a normal fluorescence microscope, all of these little tags or all these little labels are glowing all the time, and the resolution of the microscope just kills you and you can't see the actual structure of what's going on.
Liu and a team of engineers at Washington University in St. Louis developed a new technique that uses temporary fluorescence, causing the amyloids to flash or blink. This allows them to see the problematic proteins in real time.
It actually used a sensor, a little molecular marker that we know can detect amyloid proteins, but not permanently attach it. When they randomly collide with the protein that we're interested in looking at, then they glow. So by having those glowing events happen whenever those little sensors encounter the amyloid protein, kind of like you can think of little fireflies that only turn on when they're right next to the amyloid protein that they're targeted to, then we can use what's called super resolution fluorescence microscopy to reconstruct a high resolution structure of the amyloid that you wouldn't be able to see with any other technique.
Liu says having a visual of amyloids is key to understanding devastating age-related diseases like Alzheimer's and Huntington's disease.
Figure out which types of structures actually are the ones that cause disease. The things that we can really see very well with our technology now are these long fibers that are the precursors of the big plaques that you might see from patient samples. We'd like to push our transient amyloid binding or TAB microscopy technique so we'll be able to actually see them interact with neurons, how these amyloid proteins actually aggregate within cells, figure out which structures actually could cause the death of neurons down the line. With conventional imaging technologies, we don't have the resolution or the speed at which to see these processes happening in real time.